our speaker for tonight, Bruijn Method. He's right over here. What to say about him? He's got his PhD in genetics from UCLA. He's a bioinformatics scientist at Zymo Research here in Irvine. He teaches at UCLA, and these are some of his current projects. I'll let him speak about them rather than me. And with no further ado, we'll turn the evening over to Michael. Before Trey.
Yep. Okay, quick sound check. Can people hear me okay? Cool. All right, so as he said, I'm Michael Weinstein. I'm a scientist at Zymo Research, just a few miles north of here, basically just a little bit east of Diamond Jamboree. And I'm going to talk today about DNA sequencing and some of the computational challenges it presents, as well as some of the principles behind it. So just to get kind of a feel for the audience, who here has any significant amount of biology experience in their background? Okay, a few people. Who here considers themselves primarily computational? Okay, kind of what I was expecting. Okay, so for the people who did not raise their hands at the beginning when I asked about biology background, I'm going to talk a little bit about biology as information flow at the beginning to give you kind of a frame of reference for these things. So, there we go. Okay, so who am I? My primary research background is actually in the wet lab, so dealing with the sort of squishware, the stuff that is actually squishy. I've done research in heart disease, lipid metabolism, skeletal dysplasia, which is a fancy term for human dwarfisms, cancer biology, and infectious disease. I also, later in my career, transitioned to some dry lab roles, so primarily computational. And for that, I was doing a lot of genomic variant discovery, looking for variants causing human dwarfisms. I did some work on CRISPR. Who here has heard of CRISPR? Quite a few people. It's been making some news. So if anybody saw something in the news at the beginning of this year about Microsoft developing an AI system to try and predict CRISPR targeting sites, they mentioned that Microsoft had a partner at UCLA, and that would have been me. I was working on genomic searching algorithms to support the machine learning. Done some work in cancer biology and cancer informatics on ovarian cancer, as well as cancer immunology for brain cancers. My main interests in biology include modeling human disease, particularly using mouse models, efficiently handling large biological data sets, which is essentially just data science dealing with biological data, scientific computer program design, and especially the practices related to it. A huge problem right now in the bioinformatics field is a lot of the software is homebrew, and a lot of that software runs into the problem down the line of, hey, this works great, let's distribute it so other people can use it. Well, it ran on the developer system, and it didn't run on anybody else's. So anybody who was here for the last talk probably remembers discussions of Docker, and I suspect Docker is going to be a big thing on the bioinformatics horizon in the future. I also have an interest in information security from both the offensive and defensive side. It started off as a little bit of a hobby, but it's kind of gained from there, and I have some roles with that now. All right, and then last one of these, stuff that I like data abstraction. At UCLA, I teach for 3 days per quarter, so just basically about every 12 weeks I go up there and do 3 days of lecture, or 3 days of workshop, and I'm helping people who are primarily wet lab biologists get a little bit more into computing, usually when they need it for a project. So that requires me abstracting a lot of computational concepts into areas into things that would be understandable in terms of biology. For this, I'm going the opposite direction. I'm going to try and abstract a lot of biological concepts into computer science analogs, especially related to the flow of information. I also really like breaking things in a controlled manner, which is why I have that interest in information security. Is essentially a lot of information security testing is trying to break things in a controlled manner. In biology, it translates to, I have a sort of a keen interest in toxicology and just why various poisons are bad for you. So what is the essence of our challenge? The absolute essence of our challenge, if I had to do it as an elevator pitch, is we got to get samples. we got to get sequence from the samples, and that sequence could be from DNA or RNA, and I'll get into what those mean in a bit. And we have to test hypotheses based on the sequence. And those hypotheses can be incredibly diverse. So an overview of what I'm going to speak about today. I'm going to talk about the basic materials of life and how these are used to transmit the information of life. I'm going to talk about how we obtain material for sequencing, and that's going to be very brief. Reading that sequence. So where the squishy stuff meets the actual hardware, and that hardware is pretty diverse in how it works. Drafting a genome. So this is when you get the reads and you don't really have anything to compare to. You have to take all these short reads. We don't have technology right now that can read from one end of a chromosome to another. So taking all those partial chromosome reads and assembling them into an actual chromosome, and then ultimately into a genome. Aligning to a genome, so that's when you have a genome assembled and you have reads from another organism of that type. You can then align the reads to that sort of known reference genome and start comparing it and looking for differences. Then I'm going to talk about a few different types of analyses that may be done, and these are going to be more for just examples to give people an idea of what sequencing is being used for. So there'll be a more static analysis, and I'll talk about why I call it that in terms of variant identification, a more dynamic analysis looking at RNA expression, and a diversity analysis looking at microbiomes, which is something that I'm very active in right now. And then I'll conclude by talking about some of the big future areas that are kind of keen interest. So starting off with the basic materials of life, or as I want to call this part, biology is information storage and flow. I'm assuming everybody in the audience has at least heard of DNA, kind of heard people say, it's in my DNA or that kind of thing. What is DNA really? DNA is just a molecule. So DNA is kind of the stable storage for biological information. It's a very stable molecule, tends to resist a lot of chemical abuse. Its basic unit is called a nucleotide, which is a base and a backbone. So its alphabet, or the bases, is really an alphabet space of 4 letters. A, T, G, and C. You'll also occasionally find an N in there. The N is a placeholder for where nobody knows what the base is. So when you're dealing with real data, sometimes the real data isn't exactly clean, and you need a space holder for it. But the basic alphabet is just A, T, G, and C. There's a few operations you can do on this information. Essentially, you can replicate the DNA, which is going to make another copy of it. This is especially useful when your cells are dividing, and one copy of the DNA needs to go to each daughter cell. There's also transcription to make RNA, which is how the information gets passed along to start becoming information in action. DNA has a backbone. So the backbone of DNA is a repeating pattern of sugars and phosphates with the bases sticking in essentially towards the middle. So if you look at this double-stranded double helix, the backbone of that double helix, of each part of the double helix, is just a repeating pattern of sugar and phosphate. Sticking into the middle are the bases, and the base is actually base pair. So DNA forms a double-stranded anti-parallel structure, which essentially means that one strand is read in one direction, the other strand is read in the opposite direction, with A pairing to T and G pairing to C. It's kind of a weak pairing, so by heating it up or by treating it with salt, you can actually make it come apart. There's also proteins that can make it split apart for reading or replicating. So the primary information layer in DNA is just its code. It's A's, T's, G's, and C's. That being said, there's an additional information layer with DNA, and that comes in the form of methylation on the C bases or cytosines. That can affect how proteins bind to different regions or how easily the DNA comes apart. Along with that, you can have modification of histones, and how many people out there know what a histone is? That is roughly the percentage I was expecting. Histones are proteins for winding DNA. So DNA I'm going to talk about in the most detail here just because it's the area really that can give kind of a good frame of reference for how other things are working. But you kind of already see here, DNA is essentially a backbone with beads on a string, or beads on a backbone, I should say. So DNA as kind of a physical storage medium, it has its advantages and disadvantages. The nice thing is it's pretty stable and it's easy to replicate, as I'll show in a little bit. It has a strong negative charge on its backbone that can make it be attacked by positively charged chemicals. And physically, the strings can get very long. So every individual cell in your body has about 2 meters of DNA strained in it. And if you were to take all the DNA in your body, string it end to end, you'd be dead for this part because you took out all your DNA, but you could actually do about 3/4 of a lap around the solar system with that string. So a lot of just physical material. Fortunately, it's only a few atoms wide. So the solution to dealing with storing these strings is to coil them around a positively charged protein. Positively charged protein, physically it's shaped almost like a tablet, like you take for medication. You can kind of see it on here. You start off with a strand, you wind it around this positively charged protein. The DNA is negative, so it wants to associate with the positively charged protein. The positive charge also prevents it from being attacked by other positively charged things. So once it's coiled around the histones, those positively charged proteins, you coil the coils, and then you coil some more, and ultimately you get a condensed chromosome. So this is where actually another layer of information can be. I mentioned before the state of its winding around these histones. This is stained DNA. These are actual human chromosomes. There's basically 23 of them, so 22 sort of autosomes or non-sex chromosomes, and then the X and Y, which can determine sex. Anybody who wonders how 23andMe got their name, that's where the 23 comes from. And we can see there's a banding pattern here. And this banding pattern is staining for different chromatin states. So some chromatin is very, very tightly wound. The DNA is just about inaccessible to anything outside of it, and it's almost impossible to get the DNA apart. Some of the chromatin is very loosely wound. The DNA is easily accessible to things that want to bind to it. And you can imagine that's going to strongly affect how that DNA is used. Basically, if it's really tightly wound, even if there's information there, the information is essentially locked away. All right, so enough with the DNA. Let's move on to the RNA. This is getting the information in DNA more into action. So RNA, it's a lot less stable. It's extremely similar to DNA, especially in how it's physically made. You'll see a couple of changes though. Instead of a T, it has a U. So instead of thymine, it has uracil. Fundamentally, the only difference is one methyl group. So very similar. Operations you can do on RNA. You can do transcription to create it from DNA, as I mentioned before. There's also translation, where the RNA gets loaded into a thing called a ribosome, and it gets turned into a protein. Backbone, almost exactly identical to DNA, with the exception that instead of deoxyribose, it has ribose. So essentially, it has the same as DNA, but with an additional oxygen atom. It's single-stranded, so this means that it can do all kinds of crazy things like pairing to itself, forming all these interesting knot structures, and all kinds of other interesting structures. So this can add to its complexity because sometimes it can structure itself in various ways to affect how it is handled. And then finally, protein. This is actually one of the hardest molecules to study. So protein, variable stability molecule. Some proteins are extremely long-lived and very stable. Some proteins are very short-lived and unstable, and a lot of unstable proteins are even unstable by design. The basic unit for this is an amino acid, and the alphabet of amino acids, there's 20 standard amino acids. There's a couple of specialized ones, and amino acids can then be further chemically modified, so really you have thousands and thousands of potential amino acids with modification. Operations you can do on a protein, you can translate it from RNA, and proteins can catalyze chemical reactions, including the creation of DNA and RNA. They can create structures, both on the microscopic level, like the skeleton within a cell that gives a cell its shape, and on a more macroscopic level, such as the cartilage in your joints. It's structured by protein. Proteins are kind of the information turned into action. They're the arms of the robot. The backbone of a protein is the amino acid peptide bonds, so a very different backbone, and actually a lot more flexible. Proteins really have nearly infinite structural complexity available to them. And it's kind of by design because these proteins need to be able to do just a huge variety of tasks. So what's the takeaway of this for the basic materials of life and kind of biological information flow? This is the central dogma of molecular biology. Information flows from DNA to RNA to protein. Occasionally, in very rare circumstances, it'll flow backwards, primarily in the case of viruses, but those tend to violate a lot of the rules anyway. But DNA can either be replicated or it can move its information to RNA and protein, and then from RNA to protein. This codec was pretty much established at the beginning of life. The same coding in me that would code for, say, a methionine amino acid will work in a bacterial cell, which is about as far as you can get from a human. Nucleic acids, which includes DNA and RNA, they're a lot easier to sequence just due to their lower complexity. And because DNA is more stable, typically you'll turn the RNA to DNA in order to sequence it. So that's actually involving a case that violates this central dogma where you go from RNA to DNA. Going from RNA to DNA is pretty much exclusively to the domain of retroviruses. So we just hijack their protein for going in the other direction. One other property of DNA that I'll mention before I go to the next section is DNA is really good for replicating exponentially. And when I say that, what I mean is, let's see if I can. You start off with two strands, they're anti-parallel. Those two strands get split apart, and you make a copy of each strand. You now have two copies of what was originally one copy. You then can copy each of those, and it goes to 4 copies, then 8, then 16, and so on. It's just 2 to the n power, but that can lead to some very rapid DNA replication. And in fact, we can do this entire process in a test tube for very short strands. Got a guy a Nobel Prize several years ago for something known as PCR. Okay, obtaining the raw material for sequencing. So this is going to be very brief, but essentially in science, everything should be reproducible. Stuff that's reproducible is considered suspect and is generally not believed. And we have 2 flavors of replicates here. There's technical replicates. So technical replicate is taking the exact same sample and running it through your pipeline twice. This is extremely useful for identifying things like if your technician is sloppy, if your machine isn't being accurate every time. In computer science, I would say this is running the same set of data through the same function to make sure that the outcome is deterministic and not random, or occasionally to make sure that the outcome is random if that's what I want. With sequencing, you typically don't need a lot of technical replicates because the sequencing technology now is pretty reproducible. That said, it's always good to have. Biological replicates, on the other hand, it's taking different members of the same group. So for example, here we have 2 black mice and 2 tan mice. And assuming those 2 mice make up 2 different groups, we have an n of 2 per group. In reality, any statisticians out there are probably cringing right now because you always want an n of a lot more than 2. But having biological replicates can point to the normal variance within the group. Biology is kind of inherently messy, it's inherently diverse, and it's very rare that you find anything where you have no variance. In fact, a lot of times in biology, you'll find there's a very wide variance, and the example I always like to give is what happens if one of these mice has a cold. So biology is noisy. You always need quite a few biological replicates to try and come to a conclusion. So what is the general workflow for dealing with sequencing? Pretty much in all circumstances, you're going to start off by wanting to break your cells open. In the case of dealing with RNA, you're going to probably reverse transcribe it to DNA to make it a little bit more stable, but that can vary. In some samples, cell lysis, or the actual process of popping the cells open, is really easy. So mammalian tissue, for example, super easy to break the cells open. Occasionally, cell lysis is not so easy. So a really good example of this is trying to break open all the microbes in a sample. Bacteria are extremely diverse. Some bacteria If you look at them funny, they'll essentially pop. Some bacteria, you can freeze thaw them repeatedly, and they come back to life. Anybody out there ever heard of listeria outbreaks? You've probably heard of them happening in frozen food. So these are bacteria that got frozen, thawed, and came back to life and were actually infectious and killed people. Listeria is notorious for doing that. E. coli, on the other hand, a lot of times you freeze it and it just all explodes. So if you're trying to get an accurate picture of the microbial composition in a sample, that can be a huge issue for you. So once you get your DNA, you're typically going to want to fragment it a little bit. That's going to depend very heavily on your sequencing method, but typically sequencing methods will have kind of a limit to how far out they can reach. So for example, Illumina, you're typically going to want sequence fragments of somewhere in the few hundred base range. You're going to attach adapters, and an adapter is essentially a dongle for random fragments of DNA. It's the thing that mediates the connection between the squishy stuff in the form of DNA and the hardware in the form of your DNA sequencer. So the adapters will handle things like getting the DNA to stick down to your flow cell, which is the actual disposable piece that goes into the sequencer. It'll handle things like giving it priming sites for that DNA replication process I just showed you. It may even barcode it so that you can tell the origin of the sample and things like that. And that's just that is literally an adapter between the DNA from your sample and your hardware. Once you've done that, you'll probably want to do some quality checks. Just make sure that your DNA doesn't look degraded or garbage or like it's all fragmented up in the wrong way. And then once you're pretty confident you have adapterized DNA of good quality, that's when you sequence it. So this is where I transition from talking about the squishware to the hardware. So reading that sequence. There's a diversity of sequencing platforms out there. Single molecule real-time sequencing, ion semiconductor, pyro sequencing, sequencing by synthesis, sequencing by ligation, and chain termination sequencing. Pretty diverse out there. However, a couple of those platforms died, and one of those platforms isn't super relevant to us today. So two of those platforms are effectively dead. I don't really see anybody running them all that much today. And the chain termination sequencing, also known as Sanger sequencing, if you look at its cost per 1 million bases sequenced, it's about $2,400, as opposed to everything else that's usually in the range of a couple dollars to a few cents. Sanger sequencing does not generate a lot of data. Sanger sequencing is actually low throughput sequencing designed for very high accuracy on a very limited sample. Typically one thing that you just want to confirm with high confidence. So that's not super relevant to us today because that does not present really any data problems whatsoever. So essentially now we have sequencing by synthesis, which is known as the Illumina method. We have single molecule real-time sequencing, which is PacBio, or I'll frequently call it nanopore. And then you have ion semiconductor, also known as ion torrent. Ion torrent I'm not going to talk about a lot because ion torrent has generally been unfriendly to open source software, and they try and keep their ecosystem very closed. They don't show up a lot in the research environment, but they're kind of there's a lot of clinical environments that favor them where they don't need kind of high-speed innovation. Illumina sequencing by synthesis, and by the way, Illumina, these guys, Illumina, just bought these guys, PacBio. So, do you have a question? Oh, okay. So the space is kind of interesting, but it seems to generally stay pretty consolidated, and it seems to stay pretty consolidated around Illumina. The essence of Illumina sequencing is sticking a piece of dye on the end of a growing strand of DNA, flashing some light at it and seeing what color it lights up, and then changing that color every time you put on a new base as you synthesize. So you get a green flash when you add one base, you'll get a blue flash when you add another base, you'll get an orange flash from another base, and so on and so on. And the machine will just read these flashes as it goes. PacBio is a very interesting method. It's essentially pulling the DNA strand through a very, very small hole. And depending on what bases you're pulling through it, you'll get different resistances to trying to pull it through. It's a little bit of an oversimplification, but that's a bit of an idea of what it's trying to do. these two methods have very different properties. Illumina sequencing by synthesis right now, it's fairly cheap, generates a ton of sequence per flow cell, typically gigabytes and gigabytes, and I think it's now even in terabytes per flow cell. Pretty high accuracy, about 98% of the time when it calls a base, it's going to call it correctly. And probably about the cheapest method out there. Although due to the fact that the environment's consolidating a little bit, it may not be as cheap as people were expecting now. The single molecule real-time sequencing, or the nanopore method, a bit more expensive, although definitely coming down in price. And it says here 99.99% consensus accuracy, and I'll go into a little bit later about what consensus and coverage mean. But essentially, 87% single read accuracy. Or in other words, if you just look at a single base that it called on a single read, about a 13% chance that it blew the call. So PacBio, definitely not the choice if you need very high per base accuracy. However, the length of the reads is absolutely astounding. I think we've gotten reads now that are in 6 figures, definitely well into 5 figures, but I think people are getting reads now routinely into the hundreds of thousands of bases on a single read. So if you need a very long read, and I'll go into why that can be important later, PacBio is excellent. If you need a very accurate read, typically Illumina is a little bit better. And of course, as I mentioned before, if you need a super accurate read on a very limited piece of DNA, typically you're going to do Sanger, which is very cheap but very low throughput. So this is where I talk now a little bit about how the sequence data is handled. And there's this frequent story in bioinformatics where I had an issue with storing some data. I came up with a new format for storing that data, and now I have 2 problems. I also hear this story a lot of times about using regular expressions, but I happen to like regular expressions. All right, so 2 formats for storing DNA sequence data, kind of raw DNA sequence data. There's the QSEQ format. And this is going to store a fair amount of data. So you see here, each line is 1 line of QSEQ format. Starts off with a unique identifier for the machine that did the run, a unique identifier for the run itself, the lane in the flow cell. So flow cells typically have 8 lanes in them. The tile number, so information on the position within the flow cell, and the X and Y coordinate in the tile. This is pretty impressive. For any individual read of DNA in a QSEQ, I can go back and I can tell you essentially what machine read it, when it was read, and where on the flow cell that read happened. So kind of cool for traceability, and traceability is very important for sequencing. The information that's more pertinent to the data, the index number, the read number. So I won't get into this too much, but you can read sequence from one end, or you can read sequence from both ends. The information presented can be useful sometimes from both ends, and sometimes the information from both ends is no more use than the information from a single end. Following that, we have the actual DNA sequence, and that's just going to be a bunch of A's, T's, G's, C's, plus either N's or dots, depending on the specific machine, to tell you that it couldn't read a base there. The last one is this, which is this set of basically a bunch of weird characters, and that's a quality string. And the way that works is the characters represent their ASCII values, and there's a table for converting that to what's called a Phred score. And a Phred score is sort of an exponential score for confidence. Typically goes 0 through 40 for raw sequence, with 0 representing essentially 0 confidence. It wouldn't have even called a base there. A Phred score of 10, 90% confident. Phred score of 20, 99%. 30, 99.9. And 40, 99.99. So a Phred score, I never actually calculate them out. I just generally go by kind of those guidelines. So for example, if I see something got a score of 30, I know the machine thinks that there was about a 1 in 1,000 chance that it made a mistake on it. So really 2 important layers of information: what base was called, and how confident was the machine in the base that it called there. The last one is just the machine's internal code for whether or not it passed quality control. This format died. although there's still people who hold on to it very dearly and it still sees some use these days, but essentially software expects the FASTQ format. FASTQ is more popular, I think in part because a human looking at it has a much easier time looking at it. So each read here takes up 4 lines, so they're just split up by a new line character as before. The top line has all that housekeeping data on where it was read, when it was read, the index, all that other good stuff. The second line is the sequence itself. The third line is usually going to have a plus, and then it may either have nothing, or it might have some notes by it. But generally, you won't find data on the third line. The fourth line is the quality string, and it's the same encoding as I mentioned before. Okay, so this is where I get from the hardware now into the software. So this is the part I think everybody probably really wants to see. So what's the challenge here? We need a reference genome. For pretty much any organism we want to study, one of the first things people will try and do is come up with a draft reference genome for it. We don't have technology right now to read from one end of a chromosome to another, unless it happens to be a very short chromosome. As I mentioned before, PacBio or nanopore technology, you can read a few 100,000 bases. Typically, a whole chromosome is going to be in the millions. So having no technology that can reliably read from one end of a chromosome to another, we have to figure out a way to assemble these short reads. It makes this a little bit harder because some of these chromosomes have repetitive sequence. And as I mentioned, longer reads are becoming more possible with nanopore technology. So what are our solutions here? One solution is basically what I call cribbing, or assembling to an existing reference. So let's say I have I have the genome right now for the Irvine brown pigeon. I want to assemble the Irvine brown pigeon. Somebody I work with has already assembled the genome for the Santa Barbara yellow pigeon. I can use that as kind of a guide to put together the new genome. Starting completely from scratch, this turns out to be a De Bruijn graph problem. So this is probably going to get more hands. Who here is somewhat familiar with De Bruijn graphs? Oh wow, I was expecting more than that here. Okay, well the good news is I kind of give an idea about it here. It's essentially taking a fragment of each read and looking for its overlaps, basically taking a sequence and then looking for where it overlaps except for one base, and building up a graph moving between these. And then coming up with a path that basically goes through here and hits every node along the way, the minimum number of times possible, will give you a draft genome. As you can imagine, for something the size of a genome, this is very complicated, and the amount of computing resources required to assemble a draft genome with no template, using that cribbing method I mentioned before, it's pretty impressive. The Velvet assembler, you need a system with 2 terabytes of RAM, and it's going to take, I think, a couple days. A program called Abyss starts incorporating parallelism, but you're still looking at about 96 hours with a lot of cores. The fastest one right now is SOAP de Novo, and that'll do in about 40 hours, but still the computing resources required for assembling a draft genome are just astounding right now. Unfortunately, I think it is just inherently computationally intensive, and I don't think we're going to see a day where this doesn't require a lot of computing resources. I can't imagine there's going to be some major hack somebody finds for this. So there is a weakness in the assembly process. Looking at this sequence here, I'm going to highlight these. Basically what we have here is we have some sequence, and then we have this long repeating stretch of GGC, GGAA. And what will happen is, if we can't read all the way through that repeating sequence, you start getting a graph that looks like this. You have one end, you have one end, you have the other end, and in between them, you have this sequence that you know is repetitive, but you don't know how many times. So is this a 1,000,000 base block? Is this just a 100,000 base block? It's hard to say if you can't read all the way through it. This was a huge problem with genome assembly before. Having nanopore now is helping a little bit because now we can get through a lot of these very long stretches, so at least now we can say things are, now we can at least say this is a block of at least 100,000. Or occasionally we can read from one end to the other and say, we have a pretty good idea now what the size is supposed to be, but this is one major weakness. Something that gets even nastier is we have two different sequences here, but both of them have this stretch that's identical in the middle. So do we have a lot of this kind of scenario in the human genome? We have a ton of it. We have things called ALU repeats, which I believe are just old viruses that integrated and are essentially just scattered throughout our genome. Line and sign elements, but all these repetitive elements just kind of scattered. This makes genome assembly also a real problem. So with this one, you'll wind up with a graph that looks a little bit like this, where you have these two elements, you have this chunk in the middle, and if you can't read through it, you know you have these guys at the end, but you don't know exactly which one goes where. So this also can become a serious problem. Again, longer reads make this problem tend to go away. All right, storing reference sequence. For storing reference sequence, pretty much the standard right now is what's called a FASTA file. It's a standard flat file, it's text. The formatting is just really simple. You have a forward arrow, or a forward caret, or a greater than symbol, whatever you want to call it. You have the name of the sequence, such as YNEN, whatever that means. And then you just have a block of sequence after it. And that's just the format. That's very, very simple. So a couple problems with this format. We know that the human reference genome and all other reference genomes generally don't match anybody. There is not a single human walking around on the planet Earth who has the human reference genome sequence in them. Because it's only a reference. It's a general idea about what the human genome should look like. We know some positions of the human genome are invariant, and we know this because we know there's certain positions where if you change one letter on one copy, it's death. It's incompatible with life. We know some positions are highly variable. These are the kind of positions that 23andMe looks at. These are the kind of positions that police like to look at if they're identifying a suspect. They're highly variable, they don't affect much. So these contribute a lot to human diversity. And because of this, the idea of a graph-based genome has been proposed. So I showed before kind of a graph for assembling the genome, but there's also the idea of this graph-based genome where you can have multiple potential paths through it that kind of cover a little bit more about standard variants you'll find. Because along with single base changes, the human genome has variants in the forms of insertions and deletions, where you either gain or lose sequence in a region. And those are much harder to understand sometimes. All right, so let's say you have a reference genome to work with. That's going to come down now to aligning. So let's say you have, for example, the human reference genome, and you have a patient show up. So we have a big collection of reads from an organism with an assembled reference genome. We need to figure out where in the genome each read came from. So I mentioned before, you're randomly fragmenting the genome to sequence it. Did I mention anything about we know where in the genome these reads came from? Because we don't. So we have to figure out where in the genome each read came from. This is actually a really fascinating challenge. I'm going to walk through a few different methods of this because this is really one of the more fascinating things that's been done, and it's really a kind of a big achievement that people have pulled off in the bioinformatics field. So typically if I said, I need you to align this short sequence to this long sequence, the first approach everybody will go to is what I call brute force alignment. It's saying, take the short sequence, slide it along the long sequence one base at a time, and count the matches. It's the most obvious solution. So here we see we go along. At the first position, we have 5 matches. At the second position, we have 5 matches. We're getting an idea here that you get a certain number of matches just by random chance. Keep going, you get a few more matches. Keep going, hey, you get a lot of matches, but still within the random chance area. Eventually, you get to a position where it matches perfectly. Except you still need to keep going across the genome because you need to make sure it doesn't match perfectly anywhere else. So, what does this look like under the conditions that kind of exist within a standard sequencing experiment and a standard human genome? I'll make a couple assumptions here. I'll assume the reads are 50 bases long. I'm going to assume there's 3.3 billion bases in the genome. That's roughly what you get with a human genome. And about 50 million reads from the experiment, which is actually what you used to get from an experiment. Now you get a whole lot more. I don't know how to pronounce this number, but I really stop caring when it says 10 to the 18th operations. That's a lot of operations. In terms of computation time, if this is completely optimized, where I don't think you could actually have it down to one operation per comparison. In terms of computation time, about 4.125 billion seconds on a computer that can do about 2 gigahertz, so reasonable computer. You'd be looking about 130 years on single core. And I haven't even talked about how we're dealing with insertions and deletions yet, which you have to be aware of when you align. So, 2 big questions. Number 1, how to deal with insertions and deletions. I'm going to bring that one up first because it's actually a pretty simple solution. Question number 2, how do we get things aligned before our grandkids try and figure out what we were doing in the first place? So dealing with indels turns out to be fairly simple. You use what's called seed-based alignment. So I'm going to take this perfect match I had before, and I'm going to arbitrarily drop 2 bases from the middle. So I just created a deletion. And if you look at it now, it doesn't match up very well there at all, even though I just showed you that's where it came from. So I'm going to break this up into 3 seeds, and these seeds are basically, I think about 9 bases each, a little bit longer maybe. And I'm going to take these seeds and I'm going to align them individually. So aligning the first seed, I find a perfect match here. Aligning the second seed doesn't really match where it's supposed to go. And we know this is prior information, but you really wouldn't know this in a sequencing experiment. It matches kind of okay here. It matches equally well over here. So we don't really know where this thing goes. Taking the next one, matches perfectly there. Doesn't match nearly as well anywhere else. So now we can kind of see how this would go. And then at that point, you can do a local alignment and figure out how these things really belong, and ultimately figure out that you have a 2-base deletion in the middle. Could this be scaled up and modified to become an alignment method of its own? Totally. In fact, seed-based alignment is used in something called blast search, which is how you actually search a sequence against a huge diversity of organisms. If I pull random DNA sequence out of the ocean, which people have done, and I want to find out what organism did I just grab, blast search is what I would use, and blast search is based off of, among other things, seed-based alignment. So how about scoring and alignment? As I mentioned before, a common theme you'll find in a lot of these methods is you won't just get back a call, like you won't get back a base call or an alignment call, you'll get back a confidence in the call. So an example here, what would score better? We have one alignment with 2 bases, with 2-base deletion. We have another alignment that's a perfect match. Obviously the perfect match is going to be a much higher confidence. Here, it's giving some examples of where you're looking at the best match and the next best match. So here, the best match is basically 2-base variation versus a 3-base variation. And then the one below, you have a 2-base variation versus a 5-base variation, which is the next best alignment for that sequence. Obviously, you're going to be more confident when your next best alignment is a much worse alignment. You're going to be much more confident, yes, this sequence definitely comes from this place. You may have a more complicated situation where you have one alignment that says this aligns here with a 2-base deletion versus it could align here with 3-base mismatch. This is a very complicated situation. There's a couple of things that would need to be taken into account. In theory, you'd want to take into account your sequencer itself. Some sequencing methods are more likely to produce insertions and deletions as sequencer error, although you really don't see those ones anymore. Most of the time, sequencer error is likely to come in the form of a miscalled base. You're also probably going to want to look at the confidences behind them. So maybe those 3 mismatches are low confidence, in which case you can say, yeah, there's 3 bases that don't match, and they're actually bad confidence calls. The other thing is, typically for confidence scores, any insertions or deletions are going to give you a big hit to your confidence score. And there's a good reason for this. If I didn't take a big hit to my confidence score every time I tried to open an indel, I could align anything anywhere. Because I could just put in enough insertions and deletions that anything could go anywhere. All right. And to just give an idea for one of the algorithms that is actually, or that is the basis for how we do our alignments now, I'm going to introduce a fictional store called Mama Mab's Panama Bananas. Why did I come up with this ridiculous name? Much like DNA, there's actually a fairly limited letter space in this. You'll also notice there's a lot of repeating letter elements here. You know, you see the ANA and the, you'll see the ANA appear a lot. The AMA appears a couple times. So a lot like DNA, very limited alphabet, and quite a few repeating letter motifs. So for convenience sake, I'm going to just condense it all to get rid of the punctuation, get rid of the spaces. If I want to align the AMA to it, there's a couple places, and I can just do this by eye. ANA, do that by eye. But if I want to do this computationally, and I want to do it on a larger scale, I need something a little bit more sophisticated. In this case, a tree structure helps. So I'm going to build up what's called a suffix trie by taking each of these and putting it essentially into a tree structure. So I'm just going to keep cutting off the first letter and sticking it on this tree. So, and then going back actually, so here I have ANAS, which it turns out I already have ANA in there. So I now have a branch point. It can go ANAN or ANAS. Then there's NAS, well I already have N on there, so I have a branch point. There's AS, well I already have A, so now I actually have 3 branches off the A. And then there's just S, which is the last letter. This is called a suffix tree. Or sorry, this is called a suffix trie. As you may have noticed, if I have a B, there's only one position I could be starting in. If I have a B, I have to be starting at the beginning. So everything after the B on that branch of the tree is superfluous. So this is how you go from a suffix trie to a suffix tree, is just you basically prune off all the superfluous branches, which is everything after the first letter on a branch. So this is very good, but it can be kind of complicated and large to store in a computer's memory. The reason I say this is it works, again, it works pretty well. So if I want to match NANA, I just really have to go NAN, and at that point I know I'm locked in at position 2. So I've gone from one operation per letter of short read times the length of the genome to 1 operation per letter of short read, plus the generation of this tree structure, and presumably this tree structure I can keep forever and keep reusing. So again, very good. This can be very complicated and large to store in memory, so it gets unpleasant there. So again, pro, alignment operations are now basically 1 operation per letter of read. It's front-end loaded, you have your tree made. Con, on the scale of the human genome, these things are horrible to store in memory, and they're also kind of painful to make. The hack is the Burroughs-Wheeler transformation. Anybody out there heard of Burroughs-Wheeler transformation? Okay. I saw one hand besides Jeff Basine, which is actually one more than I was expecting. Burroughs-Wheeler is kind of this sort of bizarre text transformation. It effectively sorts every letter by its surrounding letters. It's still effectively a suffix tree. It's a bit more, it adds some complication to it, but it makes it a lot more efficient for storage and search. It's really effective on sequences with limited alphabets and a lot of repeating character motifs. Very effective at compressing DNA sequence, especially things like human DNA that have a lot of repetition. This and seed-based are kind of the two main methods that you'll typically find being used for alignments in production, for genome-wide alignments. Too complicated to walk through in this talk, but if anybody actually wants to know how it works, let me know. Who out there has used bzip? Anybody know what the B in bzip stands for? Burroughs-Wheeler. So bzip does very good with DNA sequence. All right, once you have your alignment done, you get back the SAM format standard. You get back something usually in the SAM format standard. This is kind of nice. There can be only one. So the SAM format is pretty much what you get back aligned DNA reads in. You may do other stuff with them and move them into other formats to hold other pieces of data. But in general, if you're getting back just aligned DNA sequence, it's going to be in the SAM format. And there can be, and in this case, there is only one format that I see being used widely. I say sort of here because a lot of times people who get back DNA sequence will actually get it back something called the BAM format. BAM format is just a binary compressed version of the SAM format. That's really all it is. There's no information gained or lost. It's just compressed. It's also indexed, so you get a second file. It's usually your BAM index. But basically, SAM files, easy for humans to read. BAM files, easy for computers to read. The information between them, there is no loss or gain between them. It's just who can read it more easily. Let me see how I'm doing on time here. Okay, almost to the end. All right, and then I'm going to talk a little bit about a couple of just different applications here. So this is going to be kind of giving an overview. Static analysis, identifying variants. A human's genome is pretty much set at birth. That should not change over their life very much. If it did, you'd accumulate mutations like crazy and you'd get cancer repeatedly and probably die of God knows what. So the challenge here, identify variants with potential clinical significance based on the genomic read data. And I know your name Bruijn, or works at Ambry, which I think is pretty much a description of their entire business model, or at least a good chunk of it. Sequencer, so a couple of challenges that come in play here. Sequencers all have non-zero error rates. They're not perfect. Normal human variation is common at many sites. The human genome is about 3.3 billion bases long. So what you're essentially looking for here is a very short needle or a very small needle in a very large haystack where some idiot also scattered random pieces of wire. It's a challenge. So what are our tools for this? We have a reference genome. We can have variation data from many ethnically diverse individuals, and there's been quite a few projects putting this data together. Especially the Broad Institute in Boston deserves mention just because they came up with a really excellent data set that made life easier for a lot of people. We have the ability to sequence family members. So if we have a child with a skeletal disease and we have two parents who don't, or maybe one who does, we can sequence all of them and see if the variant we're looking at tracks with our hypothesis. Obviously, if we see a variant in the child with a skeletal disease and we see that variant came from the parent without skeletal disease, we're probably not looking at the right variant. We have error modeling. So especially if you're doing Illumina, the error models are pretty well known. So we know, for example, Illumina is going to miss about 2% of its calls. So if we see 2% variation at a site, it's probably not a mutation, it's probably just a sequencer error, especially if it was called with low confidence. Plus we have something called coverage. We're not going to sequence everything just once, we're going to sequence everything several times, or at least our regions of interest, we're going to sequence them several times. So everybody here has 2 copies of their genome, one from mom, one from dad. If you just sequence every base once, you will only see one of those copies at any given location. If you sequence every base in your genome twice, at about half the sites you'll see a read from both copies. At half the sites you'll see reads from one or the other. So still not very good. Once you start reading about 10 times or more per site, and especially once you start getting up to 20, just about every site you'll have sampling from both copies, both mom and dad's, and you'll have enough where if the sequencer made an error, you'll have a good idea that that might just be a sequencer error. So an example of reads at a site, this is with no variation. And this is typically what we want to see. We want to see kind of random starts and ends. We don't want to see two reads that perfectly overlap each other. Reading each base multiple times is increasing our depth of coverage. It increases the chance, again, of picking up both chromosomes. High depth of coverage, again, makes us able to monitor our errors. So let's say, for example, here I have a read of a T where there should be an A. Is that a sequencer error? Quite possibly. Now let's say that at this site, half my reads are T when they should be A. Is this a sequencer error? No. This actually matches perfectly with the idea that this person got one copy with an A there, one copy with a T there. Is it clinically significant? I don't have enough context to know. Dealing with insertions and deletions, and here I'm going to actually abstract away from the sequence and go just to kind of these bars to show. We definitely have a deletion going on here, represented by that black line. But we can see here all the reads don't exactly agree on where it is. And this is because aligning each read is an iterative process. It's actually nice that it's an iterative process because when you run it on a Burroughs-Wheeler aligner, you can multi-core it really easily, and you get a linear boost with multi-core. Information gained during previous, basically you don't use previous or subsequent reads to help align these things. So the solution to this problem is after you finish your alignment, start using it as prior information to improve these things. Essentially saying, I'm going to look at this, I'm going to say, okay, well clearly all these reads here, I can really account for them by just this deletion pattern. This is an improvement. This isn't a complete improvement because this still carries a certain amount of reference bias burden. Or in other words, I'm going to be biased here by what the reference genome is, which we know this person doesn't have reference. So I'm going to try and match the reference. The ultimate solution to this is take that region where we know we have an insertion or deletion and actually do a local rebuild on the genome there. We're just going to say to ourselves, I know this person's genome doesn't look like reference here. I'm going to take all the reads from this region and just do a very small De Bruijn reassembly and build them their own custom personal genome here and see what that looks like. This is done on a much smaller scale, so it's a lot faster. And it frees you from any reference bias. Static analysis starts to become dynamic when you deal with tumor variants. So cancers have mutations, that makes the tumor the tumor. Typically you'll get healthy tissue, you'll get tumor tissue. So you know what the tumor tissue started off as, because it started off as patient tissue. You can compare the tumor tissue to the patient. Sequencers still have non-zero error rates. Tumors are homogeneous and can have high mutation rates, so the tumor is going to have a lot of mutations in it already. And different cells in the tumor may have different mutations. In addition to that, you never get just tumor cells. You're always going to get tumor cells, you're going to get blood cells that are in the tumor, you're going to get infiltrating immune cells. All the blood cells and immune cells that are in the tumor are going to have normal genomes. So you never get pure tumor. And the tumor itself may have different mutations throughout. You'll gain additional copies. You have loss and gain of gene copies, and tumors themselves are undergoing a very strong, rapid evolutionary process. Basically, within a tumor, every cell is competing to outgrow all the other cells. And from outside, a lot of cancer patients are going to be on chemotherapies. Chemotherapies are just trying to poison the tumor. That provides a really strong selective pressure for certain tumor cells to take on certain properties and mutations. So this is still very much an open challenge. There have been a lot of cool developments in this area, but ultimately detecting and understanding a lot of these cancer mutations is really an open challenge for computational biology. More dynamic analysis is looking at RNA expression. I call this dynamic because this is going to vary a lot by time. It's going to vary by location. You can imagine, for example, that the RNA expression, so basically the DNA that's being sent out, turned into RNA, and then ultimately to be turned into protein, there's going to be a big difference between the pattern in say, a neuron in my brain versus a skin cell in my hand. In addition to that, it's time sensitive. So the RNA pattern in a neuron in my brain while I'm wide awake and working is probably going to be different than while I'm sleeping. So cells regulate most of their protein production through the production of the RNA for that protein. So protein is usually transcriptionally regulated. And transcriptional regulation is usually a fast process, something that happens on the order of minutes. Splicing, I'll show what that means in a minute, but that can cause a gene to have multiple transcripts. A few RNA molecules tend to be present in very high numbers. I'll talk about that in a moment. Some RNA molecules are very important, but present in low amounts. So a good example of this is the transcription factors, or things that control the conversion of DNA to RNA information. They are very important, obviously. They're also, even the important ones tend not to be present in huge amounts. And our budgets only allow so many reads per sample. So sequencing becomes a sampling problem. We know there's a pool of RNA in the cell, and we're taking maybe, if we take 50 million reads from it, then we're sampling 50 million times. Most labs I know do not do 50 million reads per cell. Most labs I know will say like, I want 1 million. So RNA splicing, what's that? That's taking the RNA, and most RNAs that code for proteins are going to have splicing done on them, where certain chunks are going to be removed during the processing of that RNA. This can be a challenge for alignment. The solution for this turns out to be seed-based alignment. So really what I described there was the cell is making its own deletions in the RNA. And with seed-based alignment, we already talked about how that fixes deletion issues. So few RNA molecules are present in very high numbers. The ribosome is the thing that converts RNA to protein. The ribosome has its own special RNA in it called a ribosomal RNA. In any given cell, you'll find about 70% of the cell's RNA is ribosomal. This problem can be very annoying because you'll lose 70% of your read depth. This is not dealt with computationally. This is actually dealt with on a molecular basis. There's different treatments available to get rid of the ribosomal RNA using different methods. Ribosomal RNA is present in both bacteria and nucleated cells, and I'll talk in a little bit, but one nice thing about ribosomal RNA is it's very similar from one non-bacterial cell to another, and it's also very similar from one bacterial cell to another, but there are big differences between the bacterial version and the non-bacterial version. So again, solution to the ribosomal RNA is just get rid of it using molecular tools. As far as dealing with low abundance molecules and limited read depth, that winds up being a mathematical problem. Essentially, with low abundance molecules, you'll have a lot of hyperdispersion where you'll just see a lot of variance there because you're taking a limited sample of something that's rare. So if you miss 1 or 2, that can have a big effect on how much of it you estimate is there. All right, and then diversity analysis. This is something that I work on a fair amount. Something that actually Zymo also works on a fair amount. Bacteria and mammalian cells, again, have ribosomal RNA, but it's very different between the two. But within the bacterial kingdom, it's fairly similar between all the species, or at least you'll find some constant regions. You'll also find some variable regions. And if you use the constant regions to start reading, and then you read through the variable regions, you've effectively set up a barcoding system for different species. And that winds up being really cool. So why do we care so much about what microbes are present in a given place? The microbes are influenced by the environment, and the microbes influence the environment. The bacteria in your gut have pretty much been shown to play a big role in digestion, probably also play a role in immunity, may play a role in cancer risk, and now there's evidence that they may play a role in things like your mood and your neurological function. So potentially very important. Also, microbes transfer between hosts. You can tell a fair amount about something's history. I know one person who actually had a microbiome looked at on her dog, and by the varying amounts of human bacteria, we could tell which dog she basically hugged more. So what's the challenge here? Identifying the microbes present in a sample with a resolution down either to genus or species. Identifying the functional groups of microbes. So for example, Trimethylamine is something that's potentially important for atherosclerosis or heart disease. And gut microbes can modify it. Quantifying, and then finally quantifying the ratios of different microbe families present in a sample. That goes back to what I was talking about before with evenly lysing microbes. So some of them are hard, some of them are easy. And if the method used to break them open is not harsh enough, you'll underrepresent the hard to lyse ones. One other cool method here is if you just get a bunch of short sequences from ribosomal RNA, you can reconstruct all the ribosomal sequences using De Bruijn graphs. And then finally, just a few quick future applications. Increasing the speed of reading will one day allow for us to do rapid diagnostics and rapid identification of contaminants. So imagine being able, for example, to rapidly identify bacteria in food and identify if there's any potential pathogens in there. Sequencing is probably going to one day be the way of the future for that instead of culturing. A little bit of fetal DNA always spills into the mother's blood, and this is actually already deployed with some limited applications, but drawing blood from just a vein in the mother, so not having to disturb a developing fetus to collect DNA and look for potential genetic disorders. And then understanding the epigenome. So I mentioned at the beginning that there's the layer of information in DNA that's set by the sequence, and there's a layer of information that exists with the chemical modifications and the histones. Understanding the chemical modification and histone layer of information is something that's being very actively pursued. All right, and with that, I will take any questions.
First, let's thank our speaker. Yeah, for tradition here that our first question goes to Windsor Brown from the IEEE Computer Society.
Right, and it's not for you, it's for the rest of the audience. So by a show of hands, how many of you are IEEE members of one form or another? Okay, oh, I got to put my hand to the Okay, thank you.
All right, and now the floor is open for questions for Mike.
Yes?
Okay, so the DNA molecule is very long. Okay? Leaving aside the length of it, how do you know that you have read the thing? Because it varies, right? It's like a distribution. So if you take a member of the distribution, you don't know which one is which, right? There's no, you have to take sample the whole thing and then, find the distribution. So how do you know, okay, I've read the genome of this animal or that that species? How do you know you have read the whole of the distribution?
So you can usually say with some probability how much you've missed. And you can usually get that probability pretty low. And in the case where you're trying to do the reads and generate a De Bruijn graph, typically there you'll be able to notice gaps. The human genome itself, which is definitely the most read genome out there, itself has known sequences that we can't place. Like, we have known gaps. And a lot of it goes back to things like repetitive sequences or things like that. But at this point, it's so well read that there aren't missing points. But in terms of being able to say that you've read the entire genome, a lot of that just comes down to kind of, when you assemble your genome, can you see gaps?
There has been a comment that the variability within a species is greater than the difference between two species. Is that true?
That the variability within a species...
Say you have two distributions, right? Two species. And what they say that within one species, the variability can be greater than the differences between two species. In other words, two species are like a chimpanzee and a human, have many similarities, right?
Yes.
And yet within the human species, there's a great amount of variability, and yet there's the distance, if I can use that word, the distance between the two species can be very small in many areas.
So that,
how do you know, when you have such a great variability within one species, how do you know that, how can you say anything when you have such a great variability when the two species can overlap a lot, greatly?
so I think that's actually a good question. The extreme similarity between species doesn't take away from our confidence a lot of times, it actually adds to it. So just as an example, this is an actual experiment I was helping with. We were getting these cDNA. So cDNA is just a copy of DNA that has the sequence for just coding a protein. And because protein is kind of the business end of the genome, it's highly conserved. So the mouse version of a protein is going to look a lot like the human version of the protein. And the sequences are also going to be very similar. And what was really amazing there was they actually had mouse sequences in there and didn't bother telling me that they had mouse sequence. They gave me the impression it was only human. The mouse sequences, the only ones that didn't align to human, were mouse proteins that there wasn't a human equivalent. And actually some experiments I've done where I want to try and remove reads that belong to humans, so I'm looking for essentially unmappable non-human reads. I've done it where I align the genome to human and I remove everything that aligns to human because I'm not interested in human for that experiment. And then I align the genome to chimpanzee. And I remove stuff that aligns to chimpanzee because that's human sequence that for some reason was not well documented in human. So yeah, in my case, we know that our drafts are imperfect. And that's, we know our drafts are imperfect, and that kind of just help, and we assume our drafts are imperfect. So that's kind of one of the assumptions going in. We also know there's certain regions of the genome that are going to be highly variable. For example, the HLA alleles, things that control our that sort of present antigens to our immune system, most variable gene between humans, bar none. So I know that in that region, I'm not going to see anything that resembles reference. In fact, the reference genome for that region doesn't have anything. It's just a bunch of N's. So we know in that region, yeah, there's tons of variability. On the other hand, for some critical genes like collagen 1 that makes up a lot of our tissues, or just about any other protein coding gene region, there's going to be extremely small variability. And it's going to look a lot, and the human is going to look a lot like chimpanzee, is going to look a lot like mouse, is probably even going to look a lot like pufferfish. May even look a lot like fruit fly. A lot of genes will be conserved all the way down. So evolution does tend to conserve things, and that's why in some regions you'll see a lot of similarity. We don't use it to make calls or anything. It is what it is. And seeing that conservation kind of tells us, seeing how well something is conserved kind of gives us a good idea of how important it is. Does that kind of get at what you're saying, or?
Well, there's two aspects. In the classical science, when you teach students to describe the results of the experiments or whatever, you say, okay, you give a result and specify the error, so a regular error or whatever. That's classical.
Yeah.
Now, when you hear about there's a trial, the criminal was convicted because the DNA was found, right? When you read the actual analysis of the DNA, it turns out that it's not what it's supposed to be. What happens is in case of a criminal, there are only a few variability within the DNA, and those few are able to identify the criminal. But those few things, it's very small. It's 1% of the DNA of the whole DNA.
Yes.
You tell me that you are convicting a criminal based on 1% of the whole DNA. Now, if I teach my student and I say, when you give me a result, tell me within 1% or 5%, right? So you teach the students to have this idea to be precise and so forth. And yet, when you do the DNA, you tell, I'm convicting a criminal based on 1% of that person's DNA. It doesn't make sense. What do you know when you say, I convicted you on 1% of your DNA?
Okay,
What is the area? You know, variability could be like 5% maybe, and yet you convict someone on 1%?
Okay, I think I know what you're asking. Okay, now I know what you're getting at now. So, as I mentioned before, there's some regions of the DNA that are completely invariant, and my DNA at that region is going to look just like your DNA at that region, is going to look just like his DNA, her DNA at that region. And that region is useless for identifying the specific human it came from. It may even be useless for identifying it as human at all, or even a mammal. On the other hand, there's some regions that are just extremely variable. They're actually called SNPs, and these single nucleotide polymorphisms, it's bases that vary in 1% or more of the people, and you find them about 1 in every 200 bases throughout the genome. Although they tend to be rarer in coding regions, they tend to be more common in non-coding regions. And a lot of people have had these things sequenced. In fact, this is what 23andMe actually looks at. And if you look at one SNP, you'll say 10% of the people have an A here, 90% of the people have a G here. So if I look at that, I can already group people into 10%, 90%. If I look at another one that's far away and completely unlinked, I can say I can divide people 50-50. Look at another one, I can divide people 60-40. If I start building up enough of these, if I say look at 20 of these markers, I can start saying that the chance of any other human having this exact set of SNPs is 1 out of greater than the number of humans that have ever lived. So you're not trying to match the genome base for base to the criminal, because that is just about impossible. Not in terms of, just in terms of technical feasibility. What you are trying to do is look for some very specific sequence that has variation, and then say, we know the variance here, we know this is a variable region, the chance of any other individual having this exact pattern is 1 out of some number. So you can say what your confidence is. And if that number is saying, 1 out of 100 billion, there have not been 100 billion humans. So at that point I can say, with that level of confidence, this DNA came from that individual. And pretty much the one exception to that would be if the person had a clone, a bone marrow transplant, or a twin. So that's actually where the confidence comes from. It's not how much of the genome you've compared, it's how many markers you have and what the probability is that there's another human that carries that set of markers. And with that, we can get to very astoundingly accurate probabilities. Does that answer your question? All right.
Entirely we talked was computational challenges, but it seemed most of the challenges were more mathematical than computational. It seemed like the computational aspects were serializable and parallelizable, but the mathematical graph theory challenges seem to be the big ones. So can you speak a little more to actually computational issues in terms of, are there any sophisticated computational techniques that are used that are other than just serializing everything?
There's a few. So I think actually what's actually funny is a lot of the computational challenges get solved sometimes when somebody's not even trying to solve a computational challenge. Like the Burroughs-Wheeler one I mentioned. Burroughs-Wheeler existed before the challenge existed, and somebody just applied it there. So,
but again, that's more of a mathematical solution to a problem than maybe I'm splitting a hair there.
Yeah, I mean, a lot of the computational challenges ultimately come down to making the mathematics more efficient. So, assembly, as I mentioned before, De Bruijn is probably the way that it's going to be done, and it's going to keep being done. But there's probably ways to improve De Bruijn graphs, and I know quite a few people who are working in computer science who are trying to improve on that.
Are there any, excuse me, any benefits of machine learning and AI in doing the sequencing and analyzing that, either predicting or kind of building the model, so to speak?
Yes. So as I was mentioning before about trying to find the individual variants, like for example, the variants that are causing cancer. One example would be looking at a read and looking at the pattern, looking at sort of the context of a read, and seeing if it looks like it might be an error made by the sequencer, so sequencer artifact, or a real variant. And that's actually something where machine learning is being applied right now, is saying, you know, this read is surrounded, you know, this variant is surrounded by 3 A's on one side and is surrounded by 3 T's on the other side. And I've seen a lot of other questionable variants that are surrounded by this pattern. So this, you know, this feature set, and it's in the middle of the read, so this feature set suggests that it's probably a bad call. There's actually programs that are doing that kind of thing right now.
So there are some models available, or
Yeah.
either open source?
Yeah, in fact, I think the National Cancer Institute has a couple of data sets available that every once in a while they'll issue a challenge on to have people try and find more accurate error calling on it. So yeah, that's one. And then the other one is, it's difficult sometimes to predict what a mutation is going to actually do to a person. So if you've seen it before, you could say the last 5 people we saw this mutation had, you know, had back problems. But if you have a mutation that you've never seen before, can you take that, go back to say a protein structure, look at the protein structure, and try and predict based on the protein structure and where this mutation happened in the protein, we think it'll affect the structure in this way, and that's probably going to cause some other disease. Those predictions tend to be very difficult, and that's kind of an active area right now. Yes?
You made a mention earlier about a company being friendly or not friendly to open source. How much of the software is open source? What's the evolution of it?
I think pretty much all the software that people are using now for genomics is open source, unless they're using that one platform. With that one platform, you upload your data to them, and it's somewhat of a black box how they go through it, which is why they're not very popular among researchers.
Cryptographer, you wouldn't trust them. So how do you trust these guys?
That particular platform I don't trust. I actually tried running some data through them once, and I wasn't very happy with it. That's why in general people are going with open source software. And in fact, a lot of times for somebody to publish their research using the software, they have to open source it. Their code has to be subject to inspection. I will say some of the code is very sloppy, but it is still subject to inspection.
You mentioned about, I'm looking more at this sort of computational aspect of, you mentioned how many hours it takes to do a particular calculation. And my assumption, in looking at it, is you're talking about a form of pattern matching, and there's a certain amount of parallelism there that's involved. So when you're using 196 cores or whatever, it takes a significant amount of time, which is obviously going to affect your ability to take the next step. How is it in practically being addressed to make those processes faster? Increase the number of cores, look at the parallelism involved. What's happening on that aspect of it? My assumption is there's a lot of cost that goes into that aspect.
There's a fair amount of cost, not as much as you'd think, because that very expensive process I mentioned, assembling a genome, that's kind of a front-end load. So you're going to do that when you make the reference genome, but once you've made the reference genome, it doesn't have to be done again. So getting out that reference genome is going to be computationally expensive, but since it only has to be done once, and typically you're going to be doing a lot of alignment to it later, the cost of actually making the reference, of assembling the reference genome pales in comparison to just all the alignment you're ever going to do against it. That's one of the reasons why aligners tend to evolve a lot faster than assemblers. Yes?
So, the problem here that you're trying to solve, as far as I can understand, is you are reading pieces of information randomly, at random length, and you're trying to find the original single sequence that was present in this case.
More or less, yeah. it's almost like a text search. You have 50 bases of text, or you have 50 letters of text out of a 3.3 billion letter document, and you know there's potentially some errors, there might be missing letters, there might be added letters, but you want to figure out where in that document that 50 letters came from.
And how do you get those pieces? Is it the nature of the reading mechanism that you cannot see the
Yes, that's a big technical limitation right now. Is some sequencing methods will give us reads of 100,000 letters, but nothing will get us to the several million we need to get from one end of a chromosome to another.
Is it because of the physical reading mechanism? The DNA
Yeah, it's just a hardware limitation we have. Somebody may one day figure out a way for us to sort of crawl from one end to the other and just read the whole thing in sort of linearly. But at this point, we don't have that technology, and it's not really on the horizon from anything I've seen.
Yes, about increasing the speed. So I know that some guy tried to use the GPU and the FPGA to reduce the processing speed as processing time. So do you have any opinion on if the quantum computer can help that?
Do I have an opinion on it? Yes, my opinion is it's awesome. To be more specific, there's actually a company called Dragon, which also got bought by Illumina.
FPGA guy.
Yeah, I remember meeting those guys when they were first getting started at a human genetics convention. I thought it was really neat. Yeah, their little device hauls ass. It's amazing. Yeah, FPGAs are very impressive. So, yeah, I mean, fundamentally, if you can get your operation onto an FPGA, it's going to probably be very, very fast. The challenge here comes with getting it on the FPGA because that makes it very hard to code. But yeah. I've seen a demo of their thing. They've gotten huge amounts of just alignment and sequencing and sequence analysis and variant calling done in a very short amount of time using their FPGA, including approaching some of the stuff I talked about earlier of a sick child comes into the hospital with a suspected genetic disease. I think this happened down at Rady Hospital. And I believe using a combination of very fast sequencers and that platform, I think they went to a genetic diagnosis within like 24 to 48 hours. Which, I mean, even when I first got started getting my human genetics PhD, something that was practically was almost unimaginable.
So how about the quantum computer? Do you have any idea about how to use the quantum computer for their assembly or?
That's going to be interesting. There's a lot of stuff I hear about that's going to happen when a quantum computer comes out. I'm sure it's going to be applied to genomics, and I'm sure we're going to see absolutely amazing things happening there. And I will probably try and check that out as soon as I get all my passwords changed. Yes?
Well, I was just going to make a statement and then also ask a question, but I was reading an article that IBM had that QX4 that they give you a quantum minute to use. Have you heard of anything like that?
I heard of something where you basically get to borrow a little bit of quantum computing to sort of take it for a spin.
Yeah.
Is that actually a true quantum computer?
I believe so.
Oh.
Yeah, it's online. Whatever you feed it, it just.
I've heard of it.
It's right back out. Yeah.
It's only 15. Yeah. Only 15 qubits.
Oh, okay.
It's an actual quantum computing machine, and you can really experiment on that.
And I was reading that they specifically did that for university and students and researchers to kind of go around it. So that's pretty cool. And then you mentioned, this might have already been asked because I walked away for a little bit, but what do you guys use to do all the computations?
When I'm at UCLA, I use a cluster computer called Hoffman. If it's not a terribly huge job, or if it's sort of trying to develop something, I use a decently powered laptop. And if it's something that requires huge computing resources, then I use AWS. And at one point I had, I think, about $30,000 worth of credit on Azure, because I actually at one point held an Azure research grant for my CRISPR stuff. But this was in the early days of Azure when it was less than easy to use.
So most applications are running on the x86, I mean, in general, right?
Yeah, pretty much everything. Well, a lot of stuff runs on x86, but actually what you'll find is kind of the lingua franca of bioinformatics right now is Python. You'll find some applications that are very high performance that will be written in C or C++, but even there you can run it anywhere because most of the time you won't get, you'll almost never get a compiled binary. You're pretty much always getting, you're typically doing a git pull, getting a make file, and just running a make build. So, you know, you can run stuff on a Raspberry Pi, and one thing that an instructor at UCLA actually kind of made fun of me for was, if I had somebody I was kind of mentoring with coding, I would frequently try and run their stuff on a Raspberry Pi to see how it performed on a limited system. This is back when there was a Raspberry Pi 2. And a lot of times if it would break it, I'd say, you know, can you justify why I can't run it on this limited system? Or why isn't it running on this limited system? It's like, oh yeah, that's true. I don't have to keep everything in memory. Or I can get rid of stuff that I've already used. So a lot of bioinformatics programs don't need insane systems, but sometimes the insanely powerful system does help. And and you know, just like anywhere else, good programmers kind of learn to recognize which scenarios justify, you know, using the full 32 gig system.
It is said that at a certain temperature, the DNA will uncoil.
Yes.
What range of temperature is that?
It's going to vary. So A pairs with T using 2 hydrogen bonds. G pairs with C using 3. So the GC bond is actually called a strong bond. The AT bond is called the weak bond. If your stretch of DNA has a lot of AT in it, it'll typically come apart at a much lower temperature. So somewhere approaching like probably 50 plus degrees centigrade. If you have a lot of GC in there, it's going to come apart at a much higher temperature. 95 degrees is considered a pretty reliable temperature to heat DNA to for a few seconds. That will just make it all come apart. So if you're asking me how you would split strands of DNA reliably, 95 degrees for about 10 seconds should do it.
We're about 10 minutes over time already, so let's have a couple more questions and then kind of break up and talk about it.
Okay, I see somebody pointing at you saying I should call on you, so.
Okay, thank you. In the realm of personal medicine, I think there's this interest in having sort of whole genome sequencing. And you talked a bit about some of this in the recent case of this child being diagnosed from a genetic perspective. If you look at the state of the art today, where are we at? How long does it take to do a full genome sequence of a human? And where is that going from a time and cost perspective? And how much of that time and cost is the mechanics of the biological, molecular stuff with the sequencer? How much of it is computational? Where does that go over time?
We're probably looking in the next few years at being able to get a full genome sequenced on a person for a few $100s within probably a matter of days. We're going to see that within the next few years for sure. The question then becomes, what do you do with that information? So the easy part is getting the sequence. The hard part is interpreting the sequence. If somebody shows up like they would previously at the skeletal dysplasia clinic that I worked with, and they had an obvious skeletal problem, we do x-rays, we come up with ideas about what they might have. We get a few ideas about what genes might be involved because we typically know what are some good candidates. In those cases, we would do what's called an exome sequence. So we just sequence the part of the genome that's ultimately turned into RNA. And we could look at all the variants there and say, hey, I suspect there's a variant in this gene, found a variant in this gene. Especially if this variant is already known to be disease-associated, then we pretty much got the case solved. It's much harder if somebody healthy walks into the clinic. Because typically if somebody healthy walks into the clinic, they probably want predictions on the future, and those can be very hard to make. You know, it's like Yogi Berra said, predictions are really hard, especially when you're making them about the future. There's a lot of services that claim they can try and predict things like your risk of heart disease, all these other things. And they're probably not entirely wrong. You know, a lot of these genes do influence heart disease, and you can say this variant will give you a 5% increase, this variant will give you a 3% decrease, and if you start adding up all these variants, you'll get a rough idea of the person's genetic burden for heart disease risk. That won't tell you, are they eating right and exercising? For a couple of these genetic issues, and this is really where personalized medicine comes in, you might say, this person really does have an overactive cholesterol production system, and if you give them Lipitor, which essentially just disrupts that system, that'll really help them. You may also find they have just a constellation of these minor contributors that you don't know what to do specifically for that minor contributor. So what the doctor's probably going to do is tell them to eat right, exercise, and if their cholesterol's high, take Lipitor. You know, the place where I think personalized medicine is really going to take off first is probably cancer. With cancer, you have the patient's genome, and then you have the tumor's genome, and a lot of times you can find very specific mutations in the tumor's genome that are going to tell you which drugs you really want to use or which drugs are probably not going to work. And that's really an active area right now, and there's a lot of people trying to use that. And to a good extent, that's even being used right now for directing therapies. So that is, I think, the current kind of leading edge in personalized medicine is cancer treatment.
Okay, one more question?
Guy in the back, because I know you've been raising your hand a bunch of times.
You mentioned Docker containers. Have you started to see some of these platforms being shifted to Docker containerization and made available as services or publicly deployable to clouds?
All right, Jeff is making a face at me now because he knows this is kind of an interesting point that I've been getting at, but a lot of bioinformatics packages, especially ones that are coming out now and are maintained by a lot of sort of foundations, have official, like, approved, maintained Docker repos, but a lot of people aren't using them. It's just a penetration into the field issue. A lot of people who are doing bioinformatics code come from a wet lab background. They don't know DevOps, and the whole thing of Docker to them is just foreign. As more people start discovering Docker, I think the adoption is going to be fairly widespread, because getting a lot of different pieces of software running can be a real bear. And the moment somebody has a problem with, well, it worked on my computer, that's usually the moment they start thinking Docker is not such a terrible idea, and that the extra day or two it's going to take to learn Docker is probably worth it. And then it gets even better when they find out that there's official Dockers for a lot of the stuff they use, and then they start using those.
All right, we'll have to draw to a close here. Let's thank our speakers again.